Welcome to this in-depth exploration of the molecular landscape of early-onset colorectal cancer. This video is designed for researchers, clinicians, and students in oncology and molecular biology, aiming to shed light on the complex signaling pathways involved in this growing public health concern. Our goal is to provide a comprehensive overview that can inform future research directions and therapeutic strategies, ultimately improving patient outcomes. Early-onset colorectal cancer, or EOCRC, is defined as CRC diagnosed in individuals under the age of 50. In recent years, there has been an alarming increase in its incidence globally. Unlike late-onset CRC, EOCRC often presents with distinct clinical and molecular characteristics, making it crucial to understand its unique biological underpinnings. Signaling pathways are essentially communication networks within cells. They control vital processes like cell growth, division, and death. In cancer, these pathways become dysregulated, leading to uncontrolled proliferation and tumor development. Understanding these disruptions is fundamental for identifying therapeutic targets and developing effective treatments. Research has identified several key signaling pathways that are frequently dysregulated in EOCRC. These pathways play critical roles in various cellular processes, and their alteration contributes significantly to the aggressive nature of these tumors. Let's delve into some of the most prominent ones. The WNT/β-catenin pathway is a highly conserved signaling cascade essential for embryonic development and tissue homeostasis. However, its aberrant activation is a hallmark of many cancers, including EOCRC. Mutations in APC, CTNNB1, or AXIN1 can lead to constitutive activation of this pathway, promoting cell proliferation and survival. The MAPK pathway is another crucial signaling cascade that controls cell proliferation, differentiation, and survival. Its activation in EOCRC is often driven by mutations in genes like KRAS and BRAF. These mutations lead to hyperactivation of the pathway, promoting tumor growth and resistance to therapy. The PI3K/ACT-mTOR pathway is a central regulator of cell growth, metabolism, and survival. Dysregulation of this pathway, frequently observed in EOCRC, can be caused by mutations in PIK3CA, PTEN, or AKT1. Such alterations contribute to increased cell proliferation, reduced apoptosis, and enhanced angiogenesis, fueling tumor progression. While WNT, MAPK, and PI3K pathways are extensively studied, emerging research highlights the involvement of other signaling pathways in EOCRC pathogenesis. These less commonly explored pathways offer new avenues for understanding disease progression and identifying novel therapeutic targets. The TGF-beta signaling pathway plays a dual role in cancer, acting as both a tumor suppressor in early stages and a tumor promoter in advanced disease. In EOCRC, dysregulation of TGF-beta signaling, often through mutations in TGF-br2 or SMAD4, can lead to impaired tumor suppression and enhanced metastasis. The p53 pathway is a critical tumor suppressor pathway, often referred to as the quote, guardian of the genome. Mutations in TP53 are among the most common genetic alterations in human cancers, including EOCRC. Loss of functional p53 abrogates cell cycle arrest and apoptosis, contributing to genomic instability and tumor progression. Notch signaling is a highly conserved cell-to-cell communication system involved in cell fate determination, differentiation, and proliferation. In EOCRC, both oncogenic and tumor suppressive roles have been reported for Notch signaling, depending on the specific context and genetic background. Further research is needed to fully elucidate its complex role. Understanding these dysregulated signaling pathways has profound clinical implications. They represent promising therapeutic targets for precision medicine approaches in EOCRC. By inhibiting these pathways, we can potentially halt or reverse tumor growth and improve patient outcomes. The identification of these dysregulated pathways opens doors for novel drug development. Targeted therapies designed to specifically inhibit aberrant components of these pathways are already showing promise in clinical trials. This precision approach aims to maximize efficacy while minimizing off-target side effects. Furthermore, these pathways can serve as sources for biomarker identification. By detecting specific mutations or altered protein expression within these pathways, we can develop diagnostic tools for early detection of EOCRC. monitor disease progression, and predict treatment response. Future research will focus on several key areas, including comprehensive genomic and proteomic profiling of EOCRC to identify additional novel pathways, understanding the interplay between different dysregulated pathways, and developing more sophisticated preclinical models to test new therapeutic strategies. Ultimately, the goal is to move towards personalized medicine approaches for EOCRC. This involves tailoring treatment strategies based on the unique molecular profile of each patient's tumor, ensuring the most effective and least toxic therapies are chosen. Comprehensive genomic profiling, combined with advanced bioinformatics, will be instrumental in unraveling the intricate network of dysregulated pathways in EOCRC. This integrated approach will pave the way for a deeper understanding of the disease and the development of truly transformative therapies.